Computer, share screen. Okay. Immunizations and vaccines. So it's a very timely chapter because we are in the midst of a vaccine creation for COVID and how that's going to, we're going to find out how that worked, which one they came up with. If you hadn't been paying attention, hopefully we'll enjoy this one. So got some a little bit of preaching to do on vaccinations, and we'll talk about your chapter covers everything from the beginning to the 2010 study on autism linked to vaccinations and how that was published and the result of that. So we're going to talk about how important vaccines are. Y'all ready? Oh, in lab today we have, we're going to be creating our immunodiffusion lab. So we'll be working from time class gets out until lab gets going downstairs, trying to get things that would normally be time consuming already put together so that we can hopefully have each of you pour gels. Still not sure exactly how that's going to look. Y'all do have heme lab today?
Yeah.
Okay. All right. So we'll keep it as normal and as best we can as far as creating. I'm thinking that we may be able to pour a gel, run a gel, and then hopefully we can keep pouring. Hopefully we have enough that we can do it 3 times this afternoon. Fingers crossed. Okay. And then we'll on Wednesday we'll be examining the results of our immunodiffusion. So that will be it. So we're going to try to put this together as best we can, putting our minds together, our heads together, to see how this is going to work this week. So chapter overview, we're going to go over types of immunity, active immunization, passive immunization, and adoptive immunity. So we'll cover how those all are classified and the differences in those. So basic, start off there with immunity, condition of being resistant to disease. Yay? Are y'all excited? Because okay, let me try to excite you a little bit more. What's going to happen at the end of the month of December? What are you going to be faced with? What do you think? Zoom, you can chime in. Y'all can unmute and chime if you got a quiet room. What are you going to be faced with at the end of December?
Is that when the vaccine is supposed to come out?
Yep, you are correct. So it's going to be probably designated for high-risk population first and work its way down. But you're about to get faced with it. New vaccine. I don't know if y'all are excited about that or not, but it's coming. It's coming. So I'm just looking at the faces in here, but the heme test must be a booger coming up, is it? Has it got you worried?
A smidge.
A smidge. Have y'all taken it yet?
No, it just opened like 30 minutes ago.
It just opened, so y'all, okay. Let's try to put that aside. Let's try to focus on our one chapter left. And I mean, we've got some other chapters, but our last chapter for us, because y'all have already seen the questions pretty much relating to this if you've taken the quiz, because they were already put into the first quiz. Yay.
I have a question. It's kind of random. Is there a way for these, like for the final, that you could combine all of those polls and make it one big one, or are we gonna have to go in individually? Okay.
Just study each one. That way you know where. Yeah, that would be it would be like one question per pool on your quiz.
Oh, I also have a concern.
Okay, sorry.
I don't remember if it was the micro or the immuno, because they're kind of similar this round. And
They are.
I think it was the micro. I got like a 98 point something.
Mhm.
It would have been 100, but the letters, there were multiple answers for like one, and I didn't know which box to put them in. So I didn't know if you could just like go in and change it.
I don't know. We'll see.
They were
But
like
98 is
matching.
very good.
Well, they were like matching,
Oh, I know it's matching. I left it in there on purpose so you can study.
but I didn't get it right because
Oh, I don't
No.
even know if a right answer was designated for that. So, yeah, I saw it when I created it Friday. I saw that. But yeah, I'll look at it. Okay. All right, so let's get back. Immunity, okay, condition of being resistant to disease. So that's what we're going to try to create at the end of this next month, is giving you a vaccination to create immunity to COVID so we can eliminate positive cases. Okay, so that's our yay goal for into the next year, that we start to see the decrease and not this projection of all, for the next month or two. Immunization, process by which immunity is acquired. So we have, I think you understand those two. There's 3 types of immunization. There's active, passive, and adoptive. And we are going to work with active. Okay, that's our first one that we're going to work with. That's going to stimulate you or your own immune system. So you get a shot, you get some kind of vaccination to mount an adaptive immune response to the antigen. So you can do this naturally, like I did, end up getting it, getting the actual getting the actual pathogen, the virus in this case for COVID, and I produced an antibody to COVID. Yay, right? So that was a natural exposure, and that's kind of where everybody has been up to now, and probably will continue for a little bit longer, is that if you do get exposed, you're going to have an active immune response, which is good, right? As long as 99% of the people that get it this way have no issues, and they get over it, and they're working their way back to normal, and they have immunity. So you'll hear all kinds of things, or I've heard all kinds of things, even on a call with even those that are in micro. I'm on a call Thursday night with your author of your micro book, Pat Tilley. Pat Tilley has had COVID just like I have. And even in that group, we were in a group of, lab professionals. Even in that group, there was a question as to how long does the immunity last? Okay. So most of us believe that once you get the antibody production, right, that you may not have it high for a long period of time, but you will definitely have it. So that's good. So we all believe that. We don't believe there's any problem with natural exposure to the infection and producing the antibodies to the antigen itself. But that's active immunity, naturally exposing and producing an antibody. We see this with group A strep, strep pyogenes. You get strep throat, you produce an antibody to it. Okay, can you get it again? Yes, right? But you're going to fight off the first infection. Administration of a vaccine is another way of getting active immunity. This is a developed vaccine to create immunity, and such as all of our vaccines that we have are pretty much active immunization, where we get measles virus after the recipient of our immunity to a measles virus if you take the measles vaccine. So you have mumps, measles, and rubella, and we should be immune to all of those. But I've given you, kind of the bumps in the roads along with vaccinations, whether it be for varicella, whether it be for measles. There can be not 100%. There can be things like hepatitis B. You don't produce the right antibody because you didn't get enough dose. So we see that, and we're going to be seeing that play out in the next few months. So get ready. Passive immunity. This is where we actually transfer the antibodies from an immunized host to a non-immune individual. And we think of this with babies. Okay, we transfer mother's antibodies, go to the fetus or the infant. The IgG goes through the placenta, IgA, example, through breast milk. And, but it doesn't last long, okay? But it does protect. So we know all of our infants have to come off this transfer of passive immunization to creating their own active immunization. So the babies, you did this when you were born, but this is a way of protecting the newborn from illnesses early. Passive immunotherapy, this is where we can actually take antibody prep, purified antibody from an immune person and transfer it. And we've heard all this with COVID. We've heard, with plasma donation, once you've recovered, you have antibodies. Those antibodies could be given to a patient to help them. Convalescent treatment is what they call it. And this is where you may take a patient in the ICU that's struggling and infuse them with antibodies from an immune host to get over to help check the virus, check it from replicating, check it from continuing to cause harm. And we definitely have seen that play out in the last few months. This adoptive immunization, this is where you actually transfer the cell. So not the antibody, which you would do with passive, and you didn't get the illness yourself to create your own antibodies. This would be the transfer of cells of the immune system, usually lymphocytes, from an immunized host to a non-immune individual. Adoptive immunotherapy, hematopoietic stem cells into the leukemia patient who received high dose of irradiation or chemotherapy. So once you knock down somebody's immune system, transferring in the immune cells to help them develop some immunity or fight off some diseases. And that's what we've always talked about with, everything we've talked about. If you got an immunosuppressed individual, none of this makes sense, right? Because they can't produce their antibodies to any of those vaccines or any of those things. But this would help that patient. So we kind of hit on this already, but what are the advantages or limitations to active immunity? Active is great because it induces long-term protection. The one thing it's not great at, it takes time to develop. So even at the end of December, when they start vaccinating patients for COVID, it's going to take time for that patient to develop antibody. So it's not going to be this, get the shot, go to the restaurant and say, here I am, COVID, give me your best shot, right? We don't want to do that. So we still need to be careful even after the vaccination to give your body time. Some of the things we're going to be looking at in this chapter are what are some of the problems with vaccinations. So we're going to have to deal with that. We're going to have patients that are going to be reporting issues. We're going to have reactions to the vaccination. It will be very, very difficult to sway public opinion toward a vaccine if there's problems. If people start reporting this, that, and the other after getting the vaccine. But the one thing I'm not concerned so much about is, I don't want to get COVID. If I take the vaccine, I'm going to get COVID because it's, for 99% of the population, it's no biggie. Okay. So I don't think it's going to be scary to think, I don't want to take this vaccination. Like if somebody came out with, say, name something you're scared of. What are you scared of? Rabies, Ebola, comes out with one of those vaccines and they're like, well, we're not really sure we attenuated it enough or we killed it. So it's kind of a live vaccine. Are y'all up for that? No, we wouldn't be up for that. But this one has, I think what I've heard, this is a recombinant vaccine that's coming out. We're going to talk about how that happens. So you're going to get some up-to-date, real-time, nothing that you can tell your friends that everything we do in lecture seems about 20 years in the past. We're going to hit you right on the day today. Passive immunity, Immediate protection. So that's great about passive because you get the antibodies right away. So like if you got, say we had what? Needle stick. You had a needle stick from a known HIV patient or a known hepatitis patient, and you weren't vaccinated against those. Passive immunity would be very well served right there. Let's give you a dose. Let's give you a dose of antibodies because that virus has been introduced into your body. And if you can give the antibodies a chance to immediately chase it, immediately look for it, immediately come in contact with it, to get the immune system revved up and going early, then you may not have the disease. We catch it like that. So immediate protection for passive immunity. It is only temporary. Memory is not produced, and it can induce hypersensitivity. So that's one of the things that we're going to talk about with the early. With passive is hypersensitivity. So you may react. Adoptive can transfer cell-mediated immunity. Patients' own immune cells must be depleted and possible rejection because it is a transfer. It is a transfer of somebody else's cells into you. So what is a vaccine? Vaccine is an antigen suspension derived from a pathogen. So everything that you've heard since day one since it's been a novel vaccine, a novel virus with COVID is that we need to know its makeup. We need to know its gene pattern, the RNA pattern. We need to know the proteins that it produces because we're going to use those against it. We're going to use those to create a vaccine that induces our bodies to become immune to the actual virus. So a little history, backtracking a little bit. History of vaccines have been around, since the Greeks. Okay, so your first thing it says is in Greece in 4030 BC had an idea that people were catching smallpox. Okay, they were dying. They called it the speckled monster. So did you catch the speckled monster? And if you did catch the speckled monster, you're probably going to die. Okay, so they asked others affiliated with deadly disease. These observations were produced. What they came up with was this variolation. Okay, and you see that with ancient China. Variolation is to take parts of the pox itself, kind of scrape it and make a powder out of it, and then blow that into somebody's face or throw it into their face and they breathe it in. So basically you're vaccinating them with a dust of virus particles from smallpox, okay? And a lot of people didn't get smallpox, a lot of people survived, but some people got smallpox from that and actually died from that technique. But that was way back in the Greek era and in the early ancient China. So that same kind of thought pattern kind of developed, and what you're seeing now is at the time over 400,000 people in Europe died each year from smallpox. Okay? And you are thinking, well, that's nothing against COVID, right? Right? So, got to put those numbers in perspective. So there was variation in this, and it started with farmers started doing with milkmaids, right? So we get the milkmaid story with smallpox because what was seen is cowpox. They were getting exposed to cowpox by milking cows, which the milkmaids weren't getting smallpox. And if they did, it was not at the level of scarring. So it was always noted, and I don't know if you know anything about men, but if men notice that certain females have greater complexion than the others, they might play with that. They may take note of that, right? So the men used the women's complexion of the milkmaid to note that they weren't getting smallpox. They weren't getting scarring on their face from a smallpox outbreak. So that was farmers, and then Edward Jenner, who really gets the credit. But this was, as I said, this was been going on since like the Greek era. Jenner, what he did was he actually injected a patient from one patient to the other. Okay, so they had a smallpox positive patient, and he injected another patient with it, and basically became the first vaccination. And he named it Vaxa, and this was kind of fun for me to read, was Vaxa means cow. Okay, I don't know if y'all saw that when y'all were studying for the lecture. But vaccination, Vaxa in Latin means cow. So that's how Vaxa, if you want to know how we named it, vaccinations, Edward Jenner gets that credit. So more of the history from the 1880s, Louis Pasteur, I think you all know him for pasteurization. He used an attenuated organism to develop vaccine against chicken cholera and anthrax and rabies. And attenuation just means you use the bacteria or the virus itself, you weaken it by chemical treatment, you grow it in different temperatures, and repeat it in the vitro passage in cell culture. So what you're doing is you're trying to destroy not the whole bacteria or the virus, leaving enough there that it elicits an immune response, but you're trying to kind of weaken it to the point where it actually doesn't cause the illness. So attenuation was used kind of like an inactivity. So you kind of inactivate the live organism. So then we move into the 1900s or the 20th century, known as the golden age of vaccine development. We got plenty here. A new method of attenuating organisms, so that helped us with TB and typhoid fever. There was a development of toxoids, which are inactivated bacterial toxins. We use that for diphtheria and tetanus. Attenuated viral vaccines, polio, measles, mumps, rubella, varicella. Production of a glycoconjugate, which is a purified bacterial polysaccharide linked to a protein. And then our first recombinant. This is where we're going to spend a little time talking about how that works, because these are genetically engineered vaccines used against Hep B. So hepatitis B, I want to say, was developed in the 80s. I think that's where we came up. The HIV in the 80s, I want to say it was
1986.
1986, thank you. I knew it was somewhere around my graduation year. So beyond that, so what we're in now, beyond the 20th century, so now we're in the 21st century, right? The centuries go in front of the year, so we're 20 years into the 21st century, right? So more live attenuated vaccines, influenza, rotavirus, herpes zoster, multivalent glycoconjugates. This is good for pneumococcus, meningococcus, recombinant vaccines to prevent cervical cancer, HPV. We've seen that one. My kids have both been vaccinated with that one. And then new technologies to develop the next generation vaccine. So basically things that we hadn't come up with a vaccine for, we're always developing. When we talk about polio, we talk about Edward Salk and polio. All right, it was redesigned later after that and made it more effective. So we're always trying to make these vaccines better. All right, so let's see. Reading along, I think it was red in the back. There's one part in here I just don't want to miss. That's why I'm trying to make sure where I've got it. Oh, the numbers, right? I think that was early. I think we got past it without me mentioning it. We'll get there. I don't think we're quite there yet, because I want to talk about it. All right. Any questions so far? Doing all right on time? No questions from Zoom? All right. I think, yeah, I remember where it is now. When I talk about the research that was done to try to link vaccinations to autism, remind me to go over the numbers then, because that's going to kind of wrap it up. We'll get there. Sorry. I knew I had those numbers in my head that I wanted to talk about. Antigen prep used in conventional vaccines. Whole organisms, we're using it, right? Whole organism. So in this attenuated or inactivated, they call it live attenuated vaccines. Attenuated vaccines have been the routine use since Jenner's discovery of smallpox. Part of the organism, you can either use the whole organism or part of the organism, and we just went through those as toxoids with diphtheria, polysaccharides, the glycoconjugates, and purified or recombinant proteins. So let's look at attenuated. They use a live but weakened virus or bacteria. I think we've covered that already. The organisms are grown under abnormal culture conditions so that they are no longer pathogenic. Okay, so what you're doing is taking away the disease-causing ability of the virus or bacteria, but they're still able to stimulate an immune response. So we have this with bacterial. Mycobacterium bovis, adopted to grow under high concentrations of bile, TB, typhoid fever, Salmonella typhi, a mutated strain. But with the viruses, this is the one, probably the most famous, is polio. 3 poliovirus serotypes were given orally. Each has a unique antigen that can be identified by serological typing. Do any of you have parents that were old enough to get the smallpox vaccine? Y'all seen that one on your parents' arm? Some of them are still getting it. It was like it scarred you. So it had like a round head of a needle, and it actually hits into the arm and actually scarred the arm. And they quit giving it about the time I was born. So about 1965 was probably 66 where they quit giving that. But a lot of people have that still to show a vaccination. So hopefully we won't be scarring anybody with COVID vaccine. Influenza, we're very familiar with the influenza. 2 examples of a live or attenuated vaccine here. This is the nasal mist. 2 influenza strain A's with 2 influenza strain B's mixed together. And remember, I told you that it is not recommended to do the nasal mist. So I would highly discourage that if somebody gave you an option on doing that. It's been, anybody get a nasal mist this year for flu? I don't see it as much as I used to. So I think the doctors have finally got the word out that they don't want that one.
They offered it at Valley View for my daughter.
You didn't do it, did you?
No, she got the shot.
Good. Okay. Yeah, and it's nothing that has been proven. It's just that there are stories, and these have come from a doctor friend of mine that says there's definitely issues with that one, that it could be it could actually cause harm. He thinks it's just too close to the blood-brain barrier when you do the nasal mist.
Well, I had signed her up for it so she didn't have to get the shot, but they ran out before they got to her, so I'm glad I know that now.
Yeah. Okay. Always trying to be helpful. he just told me don't ever do that one again. I did it one time and I told him that like my nose ran for the whole day afterwards. I mean, just like uncontrollable nasal drainage kind of thing. Like always had to get a Kleenex all day long, wipe my nose all day long. And he was like, you're risking, you're risking a bad deal if it moves through that blood-brain barrier. So he was very against it. So examples of live attenuated vaccines that we're familiar with: mumps, measles, and rubella, chickenpox, varicella, shingles. And you can pretty much mark out the live, okay, because nobody's getting live vaccinations like they used to. So all these have been attenuated. Advantages and limitations of live attenuated: they stimulate both humoral and cell-mediated immunity. They cannot be administered to the immunocompromised patient. I think we've hit on all these so far. Potential interference by the maternal antibodies, and then require careful handling and storage. On rare occasion may revert to pathogenic form, right? That's always encouraging. Rare occasion, right? And that's what he was talking about. He was talking about just a rare occasion of possibly causing some issues. So this is more about what we like to see. We like to see inactivated vaccines. So an intact virus or bacteria that's been killed, okay, by treatment of chemical or heat. So this is more what is a little more encouraging, or you can get the patient to think a little more, not be as scared of this one as others. The intramuscular polio, influenza, again, intramuscular, intradermal, the two influenza A strains with two influenza B strains, and hepatitis A is actually one that's been inactivated for it. The advantage here is more safe, and it can be given to the immunocompromised individual because you're not worrying about it reverting or going back and causing problems. Stimulates the humoral immunity, but little or no cell-mediated immunity. So since it's not alive, you're just going to have to rely on the humoral immunity to be stimulated. May require 2 or more booster doses to produce protective immunity, and that's common. All right, when we think about hepatitis B, it is a 3-shot series. Most of your childhood vaccines were giving over a 4-visit period, where you went back every couple of weeks and got another booster and another booster, so that we could get safely and slowly, not to make the patient have to, I just remember mine, it seemed like he got 4 shots and 2 legs and probably got vaccinated like with 18 different things, it seemed like. And off he goes. Take him home. It is scary, especially for new parents. I know we don't have very many parents here right now, so it is definitely a scary situation, because what they do is they basically hand you a bunch of color-coded CDC information and say, read it and say what you think. You're there, and they say, read these, and then tell us if you're willing to let us do this to your kid. So it is kind of scary all at the same time, and but you can do your homework ahead of time. But it's all about, hey, there is a risk. There isn't a risk. You should be fine. Everything looks, and this is where you have to just look at your pediatrician and say, are these safe? Or whatever you're doing to my kid, is this going to be safe? And they will probably all tell you yes. So, but you will get this again when you go get the COVID vaccine. You're going to read. There will probably be a book to read before you take that vaccine on all the things that they've discovered in the time period that they started working on the vaccine to the time they actually distribute it. So we got live and attenuated. We've got dead and inactivated, But we can also do subunit vaccines based on what is from the pathogen. So we can take the toxoids, polysaccharides, protein, and recombinant proteins from the actual pathogen, purify those, and give those in a vaccine, or a combination of those given in a vaccine. So when we think of toxoids, we definitely think of diphtheria and tetanus. Chemically inactivated bacterial toxoids that are not pathogenic. They retain their ability to stimulate the immune system still, and we give those in vaccination against those. So vaccination against diphtheria and tetanus. Polysaccharide, biochemically purified polysaccharide from the bacterial capsule. So strep pneumo, okay? 13 or 23 serotypes of strep pneumo. Haemophilus influenza type B, HIV. I remember everybody gets their HIV early. Neisseria meningitidis. So we use the polysaccharides from these capsulated bacteria. But sometimes we have to conjugate them with the carrier protein to induce the IgG production. This polysaccharide by itself may not induce enough IgG, so you put it in with a conjugate, carry it in. Purified protein, biochemically purified component of the microorganism. Example here would be pertussis for whooping cough vaccine. It contains 2 to 5 purified proteins from Bordetella pertussis, including the pertussis toxoid. So here's kind of this idea of where we're going. We're going to start with proteins, but this is kind of where I think the new COVID vaccine is coming from. It's the recombinant. So when you look at recombinant, this is produced genetically modified non-pathogenic bacteria, yeast, or other cells. So this is what we did for the hepatitis B surface antigen, and this is what we do for the human papillomavirus. Okay, so the HPV types. It cannot be used to produce the antigens other than the proteins. Okay, so we're not producing the things that would cause issue. So what you do is it says it takes, this is on 460. You take recombinant DNA technology has made this possible. In this method, the gene coding for a specific protein antigen is removed, isolated, and incorporated into a non-pathogenic bacteria, yeast. Okay, the genetically modified cells are cultured in large quantities, produces the desired antigen, which then can be purified by conventional biochemical methods. The first recombinant protein vaccinated was 1986 for hepatitis B. So what they're doing is they're taking the surface antigen, not even taking the virus in, okay, taking the gene from the virus, recombinating it into another organism, another bacteria, for that bacteria to produce that. So we're taking the virus's mechanism, theory-wise, right? I told you a virus invades and takes over the genetic material of another cell, produces the parts it needs, right? So we're kind of using the virus as what we know about a virus against them to create a vaccine. Okay, so I think that's what we're doing here. That's what I've heard for the COVID vaccine, is that we're taking this recombinant idea, picking out the antigens, the best antigen, again, that elicits an immune response, produces antibody, taking that, recombining it into another cell to be mass produced and then given out as a vaccine. Does that make sense? Okay. All right, advantage of the subunit vaccine: induce an immune response to the pathogenic component. Safety: avoid administration of an intact organism. Okay, so we're thinking safety all the way. And according to everybody right now, Pfizer wouldn't have made the statement that they had one ready to go if they didn't feel like it was safe. They wouldn't do that. They would have halted it or restarted it or checked up. They wouldn't have been giving us a timeline of, hey, we've got this coming out. Requires 2 or more boosters. I've already heard from Pfizer that it was going to be a 2-shot combination, so you're going to get 1 and have to come back later and get a second, just to make sure you got protective immunity. But you'll start to get some immunity up to that point, okay? Requires an adjuvant to increase immunogenicity, must be multivalent. If your broad immune response is desired. So those are some of the things with the subunit vaccine. Okay, so now we're going to switch over so we can get, I think this is pretty much a review for you. Age, you know, right? Give a vaccine to the youngest individual at risk for the disease as long as it's safe, effective for that age group. Their immune status is very important. The degree of host immunocompetence can be influenced by many factors, including age and health of the host. Vaccine composition is it alive, attenuated, tend to be most immunogenic. That'd be great, but we know those might not be the safest. So subunit vaccines tend to be least immunogenic, but maybe safer. These adjuvants that we add, substances that enhance the immune response, stimulate the innate immune system to induce release of cytokines. That's very familiar in your minds right now with immunology. Antigen deliverance system, enhance the uptake of the antigen if we add the adjuvants. And immunopotentiators activate dendritic cells to present the antigens to the T cells. So we're trying to do as much as we can to get the immune system revved up. An example, alum for an adjuvant. Oil in the water emulsion results faster, longer lasting immune response, because we don't want the body to like clear these, like get an injection and the body just jumps and it's gone, right? We got to have it kind of hanging around a little bit so that the humoral immunity will kick in. So that's all, when they were developing it, maybe it's, doesn't last as long. Maybe it doesn't stay as long, because we need to increase those antibody titers. So here is a little bit on the next generation of vaccines. New vaccine design. So just put this to today. Identify potent vaccine antigens by molecular methods. That's being done. Reverse vaccinology. So you screen the entire genome of the pathogen to identify the best gene that encodes for the best antigen. And you can try to induce a broadly neutralizing antibody. I think we've hit on all of these already. Maybe try a different route of vaccine. Some people say in third world countries, you're not going to be able to give anybody a shot. So some people kind of come up with a pill, an oral vaccine, where you take it, put it under the tongue, dissolve it. Maybe you've seen people that, they used to squirt a little bit in the mouth, have everybody line up like little birds and open their mouth and chuck, 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 right? Do that kind of vaccination. So we've seen all kinds of different ways. So here are the benefits. The benefits are that it's one of the greatest medical achievements of the 20th century, reduction eliminated many serious, this is where we're going to talk about. You did get a schedule for vaccinations. If you wanted to see how many vaccinations you got when you were a kid, those are from 462 to 463. And then if you're getting ready to have babies, then you'll know what you're up against and when you're going to be visiting the pediatrician. Okay, so here's where we're going to probably finish up today. Of course, reduction of eliminated many serious potential fatal diseases. We decrease morbidity, mortality, and community, or what you've heard of herd immunity, right? Protection extended to nearby persons who have not been immunized. So let's take the numbers. These are on 465. And I just want to say these out loud so you can hear these in case you are a person who maybe have worries of vaccinations and maybe you want to be against them. So if you look at it, what we see 2007, the overall incidence of disease in which vaccines had been developed before 1980, so the diseases that we didn't have a vaccine for, decreased by 92%, with mortality decreased by more than 99%. So vaccines are definitely making a difference. For example, before the development of the diphtheria, tetanus, and pertussis vaccine, 176,000 cases of diphtheria, 1,300 cases of tetanus, and 147,000 cases of pertussis were reported annually in the US. So after that development, 2013, we had zero cases of diphtheria, 26 cases of tetanus, and 28,000 cases of pertussis. Annual cases of measles in the United States dropped from over 500,000 before 1963 to less than 200 in 2013. And here's probably the most important thing I want to talk to you about, is that bacterial meningitis with Haemophilus has practically been eliminated. But this is where we've extended countries. Where was there's some place where it talks about feature measles. We almost, I can't remember now where it was. Basically what it was saying, I'll sum it up this way. Basically what it was saying was, we had these things under control, especially measles. Very few cases of measles. But we've had outbreaks. We've had mumps outbreaks in this state, and we've had measles outbreaks in this state, and it's all been linked to kids that haven't been vaccinated. Okay? So keep that in mind, that we were basically in a way of not seeing these ever again if everybody had been vaccinated. So this is from your book. It talks about not immunized but still healthy in the blue, the immunized and healthy in the green, and red is the not immunized, sick, and contagious. So if no one is immunized, this is where we are today, okay? Contagious disease spreads through the population. Makes sense, right? This is us right now with COVID. We need one person in the room, and the room gets it. Then we're going to see some of the population gets immunized. So we're going to see this starting in December, where some in the room are going to be immunized. Contagious disease spreads through some of the population, right? Not as much, but still spreads. And then most of the population after immunization here in the green, you see that this is going to be looking a lot better soon. So that's where we're hoping to go. All right. Just to kind of quickly wrap up, since we're running out of time, adverse effects of vaccine. I did want to talk about the one to finish up and pass. I mean, you're good with that. Benefits. Okay. What I want to talk about, since we ran out, well, we're good. I think you are good. Is this idea of in 1998. Okay, so this is from your book in 467. And I know I got two things I want to talk about, and then we'll be done. 1968, Dr. Wakefield proposed a link between the MMR vaccine and the development of autism. Wakefield and his colleagues, they hypothesized that the vaccine caused intestinal inflammation, and then that let pathogens get going in the body to the bloodstream and end up in the brain area. So that was the link of autism. It had nothing to do with mercury. It didn't have anything to do with heavy metals that were in vaccines. It had everything to do with, did the pathogens get released early, too early, and make their way to the brain to cause brain damage for autism? Okay, that was one. It's subsequently, believe it or not, if you want to believe it, I don't know, but subsequently that's been retracted because others have proved it wrong. So other people started trying to link the MMR to irritated bowel, to releasing of pathogens, and following that. So there's been this long study since then of years and years and years of watching the vaccinated kids down the road. Okay, that is one. The other that I want to point out, because this is kind of new stuff, is that these monoclonal antibodies, and this is not really per se for treatment, we do these for treatment of illnesses, and we consider that kind of vaccinations. But what I wanted you to know, there's a couple of things on the names, okay? So you get iximab, that's X-I-MAB, and you get Zumab. So these are all in the names of the monoclonal antibodies. Those all mean that they've started to be humanized. So if we have a human immunoglobulin, we go with UMAB, okay? So those are like 3 things to note with the names. So keep that in mind. So we got UMAB is a human monoclonal. Iximab is a human-derived chimeric using a mouse, okay? And then a little more is Zumab. So look on page 469 to finish up. And we got these Humira. You see these names that are on TV a lot. And you see rituximab there for my wife. She took that. That was a chimeric type of immunoglobulin. And the more important things is you see those names and you see the disease indicated for those. You see autoimmunes, rheumatoid arthritis, psoriatic arthritis, ankylosing spondylitis, Crohn's disease, plaque psoriasis. So not to say these are vaccinations, these are treatments for these diseases. Kidney transplant rejection, melanoma, allergic asthma, and then you see for my wife, it was rituximab, Rituxan, okay, which is a chimeric. Its specificity for CD20. You see what they're going after, okay, what they're trying to remove. So these are antibodies directed against antibodies, okay? So they're going after some of these illnesses. Non-Hodgkin's lymphoma, chronic lymphocytic leukemia, rheumatoid arthritis, Wegener's granulomatous. And you see this bottom one with Herceptin. Herceptin is a humanized immunoglobulin, the HER2, going after the HER2 for breast cancer. So that's kind of the new thing that we're learning, is that we are developing monoclonal antibodies to go after some of these illnesses by way of just a drug. Hey, take an infusion of an antibody, that antibody seeks out another antibody and eliminates, okay? So I'm gonna stop there. Sorry, because Dr. Walls is looking while we're taking so long. So we're gonna stop the share and we're going to end the meeting. See you in lab.